Just confirm me, am I audible to you? Everything is perfect from my side. Yeah, okay, fine. We'll start our today's class. See, today we are going to study about a cleavage and some of other parts. Okay, fine. So first thing, so let me take our syllabus. So there will be a stages, embryogenesis in vertebrates. So these are all the stages which coming under the process of embryogenesis, particularly in vertebrates. So what will be the first process? Fertilization. We know that what is mean by fertilization? The fusion of male and female gamete. Okay, that process is said to be as the fertilization, right? Now, what will be the end product of this particular fusion? Obviously, the formation of a zygote. So initially, this sperm will a haploid in condition, egg is also in a haploid in condition. As a result of this fusion, we are going to get a zygote. Okay, and this zygote, which will be obviously in the diploid condition, right? And what will be the end product? That is nothing but a zygote. And after the process of forming a zygote, and that zygote has to undergo a rapid mitotic division. So you should know it will be the mitotic division only, not other any division like meiotic and all. Okay, so this zygote undergo a rapid mitotic division. Okay, and what will be the end product? Formation of a blastomeres, this blastomeres, and after that formation of a blastula. So this will be the end product of the cleavage. And after that blastula, the process of gastrulation is going to take place. And what is meant by gastrulation? Formation of three germinal layers, that is ectoderm, endoderm, and mesoderm, due to some of the rearrangement of the cells. Okay, and this process is said to be as a gastrulation. Now, what are all the stages involved in this particular gastrulation means invagination, involution, migration, and establishment of primary germ layers. Okay, so these are all the three important germ layers, right? Now, neurulation. After the process of gastrulation, you should know that no audio. Am I clear now? Now. See, so Shankar, you have to refresh your site. Just confirm me, am I audible now? Basha, tell me. Okay, fine. Right. So neurulation means formation of neural tube from the ectodermal tissue. So you should know that this nervous system will be completely originated from the layer called the ectoderm, that is outer layer. Okay, so that will be the beginning of the nervous system development. So this entire process will be dealt with the neurulation process. Now, what will be the end product of this particular neurulation? Neural plate formation, neural groove, neural tube closure, differentiation of neural crest cells. So this will be the end product of this process, and neural tube and neural crest will be formed in the process of neurulation. And organogenesis, we know that different development of organs and organ system from the three different germinal layers. Okay, that is what a process of organogenesis is. Now, the specification and differentiation of tissue, specific cell types, organ, and primordia function. So from which layer, which organ is going to produce. Okay, that was decided in this particular process, organogenesis, right? Now, fetal development. Growth and maturation of organs and tissues, preparation for birth or hatching. Okay, so this is what a developmental of a fetus. Here, what we can identify in this particular embryo. Growth, maturation, organ development, and fetal development of specific fetal characteristic features. Okay, now here there will be the emergence of fetal structures. That is what a fetal development. And after that, maturation. Finally, it will attain its growth completely and preparation for the independent life, like They have their own organ system, they have their own physiological process, they are going to come out of the mother hatching or independence. Okay, so this is what a maturation is. Now, you should know that, see, there will be the formation and function of a fetal membrane. So, you should know that what are all the membranes in the fetus. So, the first membrane will be the amniotic membrane. Okay, the name called the amnion. Okay, what is mean by amnion? Where it is getting present exactly? It is formed from ectoderm and mesoderm layers surrounding the amniotic cavity. Okay, it is formed from the ectoderm and the mesoderm layers surrounding the amniotic cavity. And what is its function? This amniotic fluid, it maintains a stable environment for the fetus. It protects the fetus. Okay, if the mother accidentally get attached with any of the wall and all, it will be prevent from the minor accidents, this amnion. Okay, so it will avoid the friction between the stomach and the fetus. Okay, so this layer, this membrane will be act as a protective covering for the fetus, right? Now, what will be the structure? Amniotic sac or amniotic fluid. Right? What is chorion? Here, this chorion will be developed from the tropoblastic cells. So, see, you have to know there will be the two different types of cells. One is tropoblastic cells and inner cell mass. So, in this, among all these two cells, from the tropoblast cells, this chorion will get it developed. Okay, trophoblast cells of the embryo as well as the mesodermal tissue. So where it is getting surrounded? It surrounds the amnion, form the fetal part of the placenta, facilitates gas exchange, nutrient transfer, and waste removal. So all the things, with it, all the waste products is getting excreted from the fetus to the system of the mother through this particular layer, through this particular membrane called the chorion. And what will be the structure involved in the chorion? Chorionic villi and chorionic fluid, right? Now, yolk sac. So initially it forms a part of the blastocyst, involves the endoderm and mesoderm layers. It provides a nourishment. So this yolk sac is the one involved in nourishing the fetus. Okay, so from and which it forms the blood cells in the early environment. That is yolk sac cavity and vitelline duct. Right? Allantois. Here allantois will be arise from the endoderm and mesoderm. That is near the developing hind gut, that expands into the connecting stalk. So you should know the membrane and where the membrane is getting located. Okay, that is sufficient. And what is their function? That's all. And this allantois also involve their function in as a waste disposal. Okay, uric acid excretion in birds and urea excretion in mammals, and contributes to blood vessel formation. Not only this yolk sac, this yolk sac is going to form the blood cells in their early development. This allantois is going to produce blood vessel. That is allantoic cavity and allantoic blood vessels. Now, coming to the most important membrane called placenta. Here, it develops from interactions between maternal uterine tissue and the chorion. It involves the trophoblast and the maternal blood vessels. So it prevents the interactions between the maternal uterine tissue and the chorion. Okay. And what will be the important functions performed by this placenta? It facilitates the exchange of nutrients, gases, waste between the maternal and the fetal circulation. See, what are all the nutrients that has to be transferred from the mother to the baby? Everything will be transferred between this membrane. Okay, that is placenta. And what will be the immunoglobulin? What will be the antibody which is present in the placenta? Anybody remember? You can, I think you have studied in the immunology. That will be the most important questions, and you should not forget about that question. What will be the antibody present in the placenta? We have 5 different types of antibody, right? Antibody A, G, M, D, like that. And what will be the antibody present in the placenta? Yeah, this IgG is the only antibody which is crossing the placenta, right? So that will be an important question. Now, this will be present in the maternal and fetal placental tissues and umbilical cord. Okay. So, what are all the types of placenta? Here you have given with the types of placenta. So, the first thing will be the diffuse placenta. Diffuse placenta will be some ungulates where the chorionic villi spread evenly across the surface of the chorion. Here, this diffuse placenta will diffuse placenta are spread evenly across the surface of the chorion. And what will be their main characteristic is this villi is scattered over the chorionic surface, and it makes a direct contact between the maternal and the fetal blood. Typically, it is non-invasive placentation. It is mostly seen in hoofed mammals like cows and deer. So, in these type of mammals, diffuse placenta will be present. And what is meant by diffuse placenta? Diffuse placenta will be simply, we can understand like this chorionic villi will spread evenly across the surface of the chorion. That is what is diffuse placenta. Okay. Now, cotyledonary placenta. It is found in the ruminants. Ruminants means what? It is somewhat, ruminants means stomach of the stomach area of the mammal, where the discrete button-like areas called cotyledons on the chorionic interdigitate with similar areas on the uterine wall, caruncles. So this is the cotyledons is the structure related to the fetal. This is with interdigitate with caruncles. Caruncles is the structure present in the maternal, and this multiple discrete areas of the contact extensive maternal fetal interface for the exchange. So you should know in the diffuse placenta, this chorionic villi come into picture. In cotyledonary placenta, this cotyledons and the caruncles involves in transformation of nutrients between the mother and the baby, right? And this will be present in the sheep and the goat. Now, zonary placenta. This zonary placenta is found in the carnivores, where we can find a chorion forms a band or girdle around the middle of the uterus. So this chorion is there, transforms their structure as a band or girdle, okay, around the middle of the uterus. So this chorionic villi forms a band-like structure, that is we are calling them as a zonary. That will be the limited area between the maternal and fetal blood. Okay, so the only thing you should know the type of placenta and which structure come into a picture. Okay, that's all. The zonary placenta can be seen in the dogs and cats. Okay, discoid placenta. Discoid placenta is found in the primates and the rodents. Here also, the chorionic villi forms a disc-shaped structure. This is the structure which attaches with the uterine wall. Okay, this gives an extensive contact area between the mother and the baby. Okay, hemochorial type, highly invasive implantation. This can be seen in the humans, apes, and rodents. So, human beings are with the discoid placenta or the hemochorial placenta. Okay, so this hemochorial placenta found in the humans and some other primates, where the maternal blood comes into direct contact with the chorionic villi. So this will be an important characteristic features of hemochorial placenta. Kindly make a note of it. And here there will be the 3 layers separate the fetal and the mother bloodstreams. That provides the highest level of exchange efficiency. So this has to be present obviously with the humans, because humans are the most advanced organisms than any organism in the earth. So obviously this type of placenta will be very well advanced. Okay, so this makes a direct contact between the mother blood and the babies. Now, epitheliochorial placenta found in some ungulates where chorionic epithelium and uterine epithelium remain intact with no direct contact between the mother and the fetal bloodstream. So, in the epitheliochorial placenta, there will be no direct contact between the two organisms. Okay, so here the multiple layers of the tissues, the same thing. That will be different layers that makes no direct contact. Okay, so then there will be obviously less efficient exchange of nutrients and waste in case of pigs and the horses, right? Now, endotheliocorial placenta. This is also found in the carnivores where the maternal endothelium and chorionic epithelium are in the direct contact. Here, there will be the reduced number of layers. So, as it is an endotheliocorial, it has a reduced number of layers, and in case of epitheliocorial, it will be with multiple layers of tissue. As it is a reduced number of layers, there will be moderate exchange of moderate efficient exchange of nutrients and the waste products. So, in case of epitheliocorial, it will be less efficient. In case of endothelial, it will be moderate, and in case of hemocorial placenta, it is highly efficient. Okay, and this endothelial will be present in the cats. That's all about the thing. So we'll finish up to these three topics, right? For tomorrow, we are going to discuss these two topics. So coming back to the questions. Which stage of embryogenesis involves the formation of three primary germ layers? No doubt in that will be gastrulation. Now, what is the function of amnion during embryogenesis? Amnion. Protection and cushioning. Very good. This will be an important structure that surrounds and protects the embryo by forming an amniotic sac. See, if a mother experiences any maternal shock, if he making contact with any of the thing like external factors, so this is the one which prevents the baby from the mechanical shock experienced by the mother. This maintains a stable environment for the fetal development, right? It acts like a cushion. Now, in which type of placentation do chorionic villi form a disc-shaped structure? That will be discoid placenta. Kindly go through the other important informations also. Okay. Which type of cleavage typically results in blastomeres of uneven size? If you have time, kindly go through the PPT which you have listened to the class, first class. Okay. So there will be a detailed explanation of types of cleavage and also the examples also. Okay. So kindly go through that. So you can get a question from the cleavage for sure. Okay. That might be a lot of possibility. That will be meroblastic cleavage. So this is a partial cleavage occurs in eggs with a large amount of yolk. Okay, such as in birds and the reptiles. It results in the blastomeres of uneven size due to unequal distribution of yolk. Okay, so amount of yolk is a deciding factor for the cleavage, right? During which stage does the neural tube form from ectodermal tissue? That will be neurulation. Which fetal membrane primarily functions in protecting the embryo by forming a fluid filled sac? That will be amnion. Which fetal membrane involved in facilitating exchange of gases, nutrients, and waste between maternal and fetal circulations? Chorion. Very good. So you should not um, confuse it with chorion and the placenta. Okay. So that is important thing. In some vertebrates, which fetal membrane provides early nourishment to embryo and later forms the blood cells? Early nourishment of the embryo and later forms of blood cells will be the yolk sac. Very good. Which fetal membrane in mammals expands into connecting stalks and functions in waste disposal and blood vessel formation? That will be allantois. Okay, see, there will be a minute difference in the function of all these membranes. So kindly note all the keywords. Okay, this can be asked in the match the following questions, right? Which fetal membrane in humans and some other primates allows direct contact between maternal and fetal bloodstreams for efficient exchange? See, when it comes to the developmental biology, we can get a question whatever the statement exactly we have read when we prepare. Okay, so you should not avoid the question those questions from the developmental biology. Definitely that question will be easy, right? After finishing, will solve some of the PVQ also so that we come to know. That will be hemochorial placenta. Okay. Now, which type of placenta involves multiple discrete areas called cotyledons on the chorion that interdigitate with some similar areas on the uterine wall? That will be cotyledonary placenta. Which type of placenta forms a band or girdle around the middle of the uterus? That will be zonary placenta. Which type of placenta or chorionic villi is scattered evenly over the entire chorionic surface? Diffuse placenta, cotyledonary placenta, zonary placenta, and discoid placenta. That will be diffuse placenta, right? Which type of placenta involves a chorionic villi forming a disc-shaped structure that attaches to the uterine wall? that will be destroyed. Okay. Which type of placenta allows direct contact between maternal and fetal bloodstreams with only a few layers separating them? Hemochorial placenta. See, this is found in humans and some other primates. In this type, there are only three layers separating maternal and fetal. See, because of only these three layers, this maternal and fetal bloodstreams have a direct contact, and we are having an efficient exchange of nutrients and gases between the mother and the baby, right? Everybody clear? Okay. So in this endothelial, it will be in epithelial. They are having a lot of layers, so there will be no efficient exchange of gases and nutrients. In endothelial, there will be a moderate number of layers, so there will be the moderate efficient exchange of nutrients and gases. In hemochorial placenta, we are having only a three layer, right? And as it have a minimum number of layers, the exchange will be very efficient and effective. Okay. So if we start this, I think we can complete. Let's try it. Okay. The gene expression. This is somewhat related to regulation and genetic control of a development. Okay. See, this just an overview because we have a lot of signaling pathways like winter signaling, BMP pathway, north signaling, hedgehog, like that. So I give just an overview. If you want me to explain each and every signaling, let me know. Only then I can teach. Okay. If you know the signaling pathway correctly already, means no point of doing that again. Now, gene expression regulation. Here, a control of gene expression through mechanisms such as transcription factors. We know that transcription factors is very much important for the gene expression. Enhancers, repressor, epigenetic modifications. So all these things are a gene regulation, expression regulation. They are regulating the gene expression. Now, what are all the signaling pathway involved in this thing? Wind signaling, BMP, notch, and hedgehog. This is the one regulate the cell fate determination, proliferation, that is growth, differentiation, patterning during development. And what is cell-to-cell interactions? Here, the cell-to-cell interaction will be mediated by the CAM molecules, that is cell adhesion molecules and the signaling molecules. And the signaling molecules like delta notch signaling, they are the one involving that influence the cell differentiation and tissue patterning. Okay, cell to cell communication is plays a crucial role in this particular process. And morphogen gradients. Morphogen means BMP, sonic hedgehog, like that. Their formation of concentration gradient of morphogens provides a positional information where this particular organ has to be located. So that location of that particular organ and what this particular layer is going to give rise to which type of organ, their fate. Okay, their fate and their location based on their location within the embryo. That was defined by the morphogen gradient. So this is the one determining the morphological characteristic features. Now, master regulatory genes. Here, what are all the master regulatory genes? Means those are all the genes which encode for this transcription factors. We have a different transcription factors, and those factors has to be coded by genes, and that genes will be the Hox genes and Pax genes. So these are all the genes that control the expression of downstream genes responsible for specific developmental process such as body segmentation and organ formation. Epigenetic regulation, this is somewhat a post-translational modification like DNA methylation, histone modification. These are all the things regulate the pattern and during the cell division and development. And what will be the timing and sequence of events? Here there will be a precise coordination and timing of developmental events through the regulatory network and checkpoints. So these are all the things which is making ultimately the embryo has to be developed. The cell has to be divided correctly, and the organ has to be located in the proper position. And to give a perfect baby, all these things plays a crucial role, like master regulatory genes. So involvement of genes are very important. So signal. So the gene, one if one cell has to communicate with the other cell, definitely there has to be a signal. So these are all the things related to signaling. Okay. So environmental factors will be based on the nutrients, based on the temperature, and based on the level of hormones. All these will be an external factor. Okay. Now, what will be the feedback mechanism? Feedback loops, positive and negative, within the regulatory and networks that maintain a fine-tuned gene expression patterns and developmental process. Okay. So this is what a feedback mechanism is. Now, just tell me which type of molecules are central to regulating gene expression during development by binding to specific DNA sequence. Here, which molecule regulating the gene expression during the development by binding to specific DNA sequences? So it directly associate binding with the communicating with the DNA sequence. So what will be the factors? That will be obviously transcription factors, right? Which signaling pathway is known to regulate the cell fate determination and patterning during development? Which signaling pathway? That will be Wnt signaling. Okay, this is the pathway regulate the cell fate determination, proliferation, patterning during the embryonic development and adult tissue homeostasis. Okay, which mechanism provides positional information to cells by forming concentration gradients within the developing embryo? What is the answer? That will be morphogen gradients. This is the one form the concentration gradients across the tissues, providing positional information to cells, regulating their fate and differentiation during the development. Okay, which type of genes encode transcription factors that control the expression of downstream genes responsible for specific developmental process? That will be homeotic genes. Okay. Which process ensures the precise timing and sequencing of developmental events, ensuring proper tissue formation and organogenesis. That will be regulatory networks. So, okay, that's all about today. So tomorrow, I think development of frog and chick will also be get completed. So after that, after we finish this, we'll solve the PVQs from that question paper. Okay, we'll stop up to this. If you have any doubts, you can ask me now. Kindly join the class at sharp 6 a.m. Okay, because the class is going around the 30 to 40 minutes only. So you have to be um, gain something in the morning time. Okay, so this time is very effective when you learn something. Okay, is there any doubts? And just go through something about development of frog and chick. Okay, at this duration, during the minimum duration, we can't explain the all the single basic information. I can just give only the overview. Okay, so that is why. Anything else? Do you want me anything to clarify? Okay, then thank you everyone. Tomorrow the same time, morning 6 a.m. Okay, bye. Thank you.